Another of the fibrillar components of the ECM are reticular fibers. These are type 3 collagen, which forms smaller bundles than the type 1 collagen. They are also what we call argyrophilic, which means they bind silver stains. So if you are going to identify them, typically they will be stained with such a silver stain, and you can see them forming a reticulum or network throughout the tissue that they're supporting. These are the fibers that are distensible, and therefore the stroma of whatever organ they're supporting has those properties of being able to change shape slightly. The third fiber is elastic fibers. These are comprised of two proteins, elastin and fibrillin. So the elastin is the one that does the stretch and recoil, and you can see diagrammatically how that happens. In this diagram on the right, you can see the elastin is centrally located with the microfibrils of fibrillin around the perimeter of the elastin mass. And this is what you saw when you saw the electron micrograph with a cross-sectioned elastin fiber. Histologically, elastic fibers appear dark and usually fairly straight. We use special stains to visualize them. As you can see in the top image here, we used an Orcine stain. Elastic fibers also occur in layered arrangements called lamella, especially in artery walls. The large elastic arteries have numerous elastic lamina within them, and you can see them stained purple in this lower image. The final area we'd like to discuss in the connective tissue compartment is the interface between the cells and their matrix. This is a very important area because it controls the interactions between the cells and their environment. It facilitates adhesion, communication, and cell movements. Two components that you're going to find in this area are the transmembrane structural glycoproteins and the basement membrane. So the transmembrane structural glycoproteins, we've touched on these previously, include molecules such as the integrin molecules and other similar paired molecular structures such as dystroglycans. All of these molecules, regardless of how they're put together, all of these are examples here are dimers, have the capacity to link the interior cytoskeletal array of the cells to the extracellular matrix molecules. So they act as adapters between the cytoskeleton and the extracellular matrix. In such a role, they provide adhesion and traction during movements, and they also are responsible for bidirectional signaling. Now, the basement membrane is a structure that can be seen in the light microscope. This contrasts with what we were calling the basal lamina, which can only be distinguished in the electron microscope. The basement membrane consists of three layers, the two layers of the basal lamina, lamina lucida and lamina densa, and a third layer called the lamina reticularis. Now recall that those layers of the basal lamina are actually made by the epithelial cells, but the lamina reticularis is made by the connective tissue cells. So together, these three layers form something that's visible at the light microscopic level and is comprised of laminin, collagens of various types, and proteoglycans. The final topic we're going to talk about in connective tissue is how to classify them. From this little scheme here, you can see that CT proper, or general connective tissue, has several different categories, and On the right, you can see that we have embryonic and specialized connective tissues. In lab, you're going to look at connective tissue proper, or the general connective tissues, and you're also going to take a quick look at the embryonic connective tissues, because those tissues and the cells that make them are really the precursors of all of the general connective tissues. The specialized will be looked at in other units. So the features of loose or areolar connective tissue are a loose arrangement of fibers and cells, and in this arrangement, ground substance predominates over the cells and fibers. These loose connective tissues typically support epithelial layers. They line the body cavities like the mesentery. They are found around blood vessels and nerves, and underlying mucosal areas, such as the connective tissue compartment underlying the gut or respiratory systems. And in that location, they're called lamina propria. Here is a real-life example of the mesentery. This is a tissue spread, and in this you can see cells scattered about, as well as those thick pink fibers, which are the collagen, and the very thin fibers that are kind of running through, which are the elastic fibers, and the various cell types. And in laboratory, we will look at all of these and try to identify what we can. Another example of the loose connective tissue, as I said before, is that lamina propria, labeled in both of these images. And you can see it under the villi and underneath the epithelium in the respiratory tract on the right. Now, let's talk about dense connective tissue. Dense connective tissue is different from loose in that fibers predominate over the cells and ground substance. And it's actually the arrangement of those fibers which determines how we're going to subcategorize this dense connective tissue. This is specialized to resist stress, and the two subtypes are regular and irregular. So in regular dense connective tissue, the fibers of the matrix are all oriented in the same direction. And you're going to have cells also oriented, mostly fibroblasts, kind of elongated in that same orientation. You find this in places like tendons, ligaments, cornea, organ capsules, and so on. And here's a real-life example of it. And the arrows are pointing to the nuclei of fibroblasts. And all that other light pink stuff, that's matrix. That's actually collagen fibers stretched out in that one orientation. In dense irregular connective tissue, the fibers are in a mesh-like arrangement. You can see the collagen fibers in this example, and the proteoglycans and even that small black one, which is supposed to represent elastic fiber. And they are all oriented in many different directions. And you find this in areas where you have to resist stress in many orientations, such as the dermis of the skin, or capsules around organs, or the stroma of organs, and the encapsulation of nerves and muscle sheaths. Here is an example, and again, all of this pink, lighter pink stuff that you see are bundles of collagen fibers, and you can see that those bundles are sometimes oriented longitudinally, sometimes almost cross-sectionally or obliquely, in many different orientations. And this is a fairly low mag, so it's kind of difficult to distinguish the fibroblasts, but in lab you will be able to ratchet this up to a higher magnification and identify those cells. Now, you can also call connective tissue compartments that have a predominance of type 3 collagen reticular fibers as reticular connective tissue. So in this case, the type 3 collagen is oriented in a mesh-like network and is the stroma, or the supporting tissue of those organs. And you see this particularly in places like the lymph nodes and in liver and spleen and bone marrow as well. if your tissue, your connective tissue compartment has a very high proportion of adipocytes, then you're going to call that adipose connective tissue. And this is the most cellular type of connective tissue. Most connective tissues have a predominance of matrix over cells. Adipose tissue is the one exception to that. This is also a very highly vascular tissue for easy access to store the triglycerides and then to get them back out again for use in energy production. And finally, elastic connective tissue, which is characterized by a predominance of elastic fibers. And these are arranged parallel to each other in lamella or sheets. And elastic fibers are made by resident fibroblasts. And if you're talking about the arterial wall, the smooth muscle cells that live there also participate in making it. But we're going to talk about mostly just fibroblasts making them, as this is connective tissue. Now, the embryonic connective tissues are really just two types, mesenchymal and mucoid or mucus. So mesenchymal connective tissue is present only in development. These cells are precursor cells. They're stem cells that give rise to most of the cells of connective tissue. In other words, mesenchymal cells can become fibroblasts, they can become adipocytes, they can also become more specialized connective tissue cells such as osteoblasts for bone or chondrocytes for cartilage. Mesenchymal tissue contains numerous mesenchymal cells, which are spindle or stellate shaped, and scattered fibers and gel-like ground substance. So the ground substance has a high proportion of hyaluronic acid in both of these different embryonic tissues, which gives us this gel-like consistency. Mucoid connective tissue is similar to mesenchymal in that it's populated by mesenchymal cells and it has this jelly-like ECM, but here the proportion of hyaluronic acid is even greater than in mesenchymal tissue, and therefore it resists compression very well, which is important for preventing the occlusion of the blood vessels in the umbilical cord. This is a very loose kind of amorphous connective tissue. The ECM contains sparse collagen fibers and abundant ground substance. And as I said, it's populated by mesenchymal cells, which are end-stage cells that are never going to develop into anything else. So some texts call them fibroblasts. So we're going to be looking at examples of all of these tissues in laboratory.